Hey everyone, Dr. D here, and in this video we are going to be doing an exam review for Biology 2420 for Tarrant County South.
Dr. D, 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 Dr. D. Explain stuff.
So let's get started. This exam will cover chapters 1, 3, 4, and 5. Here's chapter 1. And we need to know any terms in bold, obviously. The best way to study for the exam is obviously study all of these lecture handouts here. Okay, especially terms in bold. Please learn everything the best you can. I'm going to try to highlight some of the more important information in this video, but please study every slide and learn all the concepts for your best chance to really ace the exam. So let's go ahead with this microbiology. Remember the term here. I'm just gonna skip ahead. And by the way, again, just because I skip something does not mean you don't need to study it. It just means that I use these exam reviews not to reteach everything or retouch on everything, but to go into some of the meatier parts. The parts where you may want to focus more. So again, please study all the slides. Just because I skip a slide does not mean anything. It just means that it may not be on the top of my mind for something that's a thought-provoking question. Material for a thought-provoking question. So let me skip ahead to something more meaty. All right, let's skip to here in the slides. And again, like I mentioned, please study everything, but I'm skipping ahead to here. I want you to know the 3 domains of life: archaea, eukarya, bacteria. We should know the terms helminths, fungi, which are types of eukaryotes. We should know about the size difference between prokaryotic bacterial and archaeal cells, which are 10 times smaller usually than eukaryotic cells. We should know that viruses and prions are not alive. Remember, they are not living. They are small infectious agents. We should know that it was Louis Pasteur who disproved the concept of spontaneous generation with his elegant swan neck experiment, swan neck flask. It was Robert Hooke who first observed microbes and made publications of these fungi and different microbes. But it was Anthony van Leeuwenhoek who made the early crude microscope. Remember, with his pinhole microscope that had a single lens, so it's a simple microscope. We need to know of John Tyndall and how he found that microbes in the dust and air have high heat resistance. The discovery and detailed description of heat-resistant bacterial endospores clarified the heat resistance of microbes. So essentially, he discovered endospores floating around. We need to know the term sterile. We need to know about the contributions of Koch. Remember Robert Koch? He was the one that linked a specific organism with a specific disease. So remember that he was the one who said it's Bacillus anthracis which causes anthrax, right? Linking a particular microbe to a particular disease, really solidifying germ theory. And what about Dr. Oliver Wendell Holmes? Observed that mothers who gave birth at home experienced fewer infections at the time than mothers who gave birth in the hospital. And later, it was, remember, Dr. Ignaz Semmelweis who used these observations to promote hand washing. Remember? So he was working in the maternity ward, and he promoted hand washing because he noticed that some clinicians were working in autopsy rooms without washing their hands and then delivering babies, and that was transmitting disease. So Ignaz Semmelweis was known for hand washing. Remember that? And Joseph Lister, we need to know his contribution. We need to know that he was the first to utilize hand washing and misting in the operating room with antiseptic chemicals. Remember, he would try to use antiseptics and different chemicals on his surgical instruments before operation. And Pasteur, in addition to disproving spontaneous generation, remember, he invented pasteurization. He showed that human diseases could come from infection with germs. Remember germ theory? Other things we talked about were that Pasteur showed us how fermentation works. He also contributed greatly to vaccination. So he was a real rock star. And I told you already about Koch. Established proofs. Remember, it was called his 4 postulates. He used 4 postulates in order to link a particular germ to a particular disease. Awesome. So we should know these terms: nomenclature, classification, identification. Binomial nomenclature. You definitely need to know this for the exam. Do you remember binomial nomenclature? For example, if you want to say the proper scientific name for the human species, you would say it's Homo sapiens. But remember, the proper way is that the genus name, the first word, is always capitalized, and the species name begins with a lowercase letter. Then it's not correct until you italicize or underline the word, right? So binomial nomenclature, capitalize the first word, lowercase the second word, and then either italicize or underline. Okay, we need to know that. And do you remember the layers of taxonomy, the hierarchy of taxonomy in order from domain to species? Remember I told you that mnemonic device? Dude, King Philip came over for good spices. So this is a great, mnemonic device for remembering the levels of taxonomy. And remember, there's only 3 domains of life, but then underneath domains, you have kingdoms, and underneath that, you have phylum. Don't worry about division. Then class, order, family, genus, species. Remember the principles of taxonomy. Oh, and do you remember this very important thing we touched on in chapter 1, this concept of archaea, the domain archaea being more closely related to us eukarya because we have a more common ancestor. Do you remember that? So archaea and eukarya are more closely related to each other than either is to bacteria. Okay. And that's it. That's it for chapter 1. Not too much to worry about with chapter 1, but please do review all the slides for chapter 1 to be really prepared for the exam. Be able to, like I said, talk about domain to species and order, talk about the contribution of the different early scientists and physicians. What did they contribute? What are the 3 domains of life? Are viruses alive or not? Which domains of life are more closely related to each other? I mean, these are the meaty topics that, you might see questions about on the exam, okay? So study that in detail. Try to recall those things, and I think you'll be just fine with chapter 1. Chapter 1's not that bad. All right, so let's take a quick little break time with Gizmo and Wicket, my cats, and we will be right back for chapter 3. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and continue our exam review with Chapter 3, Tools of the Laboratory. You should know the 5 I's of basic techniques: inoculation, incubation, isolation, inspection, and identification. So don't just remember these 5 I's, but be able to describe what they mean. Know, again, please know all these terms: culture, medium, inoculation, sterile. These are important terms to understand. Now, remember, there are different physical types of media: chemical composition, functional type. So remember, we talked about general purpose media, enriched, selective, differential, reducing, specimen transport, assay, enumeration. We talked about all different kinds of media, and I'll highlight those in a minute. Liquid media, semi-solid media, solid media, agar. You should know what defined or synthetic media means. Remember, this is media whose exact chemical compositions are known. And you should be able to tell the difference between that and complex media, where at least one component is not known. So complex media versus defined media. You should know that usually in the lab we use complex media. So for example, TSA plates and TSB broth are complex media. General purpose media, this is the type of, again, this is TSB, this is TSA, these are the types of media we use in the lab. Enriched media, you should know that enriched media contains complex organic substances that fastidious bacteria require for growth. You should know that fastidious bacteria are bacteria that are somewhat harder to grow because they have special nutritional requirements. You should know growth factors are specific vitamins or amino acids. You should know the difference between selective and differential media. Remember this? Selective media prevents one type of bacterium from growing and promotes another type to grow. Whereas differential media allows, of the bacteria that do grow, it allows you to differentiate between different types. So selective media, again, contains one or more agents that inhibit the growth of certain microbes and encourage a different set of microbes to grow. Whereas differential media allows multiple types of microorganisms to grow, but it helps display visible differences between different types of colonies. You should know about blood agar. Remember, we talked about that blood agar has usually sheep's blood mixed in, and this allows us to screen for hemolysins, which are enzymes that lyse or break down red blood cells. You should know that beta hemolysis is complete lysis of red blood cells, alpha hemolysis is incomplete lysis of red blood cells, and gamma hemolysis means no hemolysis occurred. So here you can see there is a nice little halo, a clearing in beta hemolytic colonies. You see there's a clearing of no red blood cells. That's because you have complete destruction, complete lysis of the red blood cells. In alpha hemolytic colonies, you can see that it's not a complete lysis. It results in partial hemolysis, which gives you a bruised effect. And then lastly, in gamma hemolysis, you can see here in gamma hemolysis, no hemolysis occurred. So let's move on to reducing media. Reducing media removes oxygen from the media and allows anaerobic bacteria to grow. Anaerobic bacteria is bacteria that grows in the absence of oxygen. And then we talked about carbohydrate fermentation media. These types of media contain sugars that can be fermented and a pH indicator to show that the fermentation is occurring. What is a colony? You should know that a colony consists of millions of the same clone, right? This is known as a clonal population. Remember, we discussed in class that each colony on the plate came from one parent cell, which multiplied numerous times exponentially. Okay, so we need to know that each colony consists of clones. Please know about isolation. You should know that the streak plate, and you guys have all done a streak plate in the lab now, and everyone did a nice job with their streak plates. The streak plate is a technique used to make isolated colonies. And then from those isolated colonies, you could make pure culture. You could subculture them. Remember that? So the purpose of a streak plate is to find individual colonies. Now, a pore plate. Do you remember we also made pore plates? Pore plate is when we add liquid agar to a plate, and later on we look for colonies and we can count colonies. Remember how you can find a pore plate, or you can identify a pore plate because there's little colonies trapped inside of the agar. So not only are there colonies on the surface of the agar, but you see these little guys right here? These are little guys trapped in the agar because you pour liquid agar into the plate, and then you allow the plate to solidify. And then there's a spread plate, right? Remember, for the spread plate, we spread it around with a hockey stick or a swab. We used a swab. So this requires spreading the bacteria around on the surface of the agar. And in this case, you should only see bacteria on the surface of the agar. You're not going to see any bacteria trapped within the agar because that would be from a pore plate. Very good. Pure culture. A pure culture is a culture of one specific species that contains only a single known species. A subculture is a second level culture from a well-isolated colony. So when you took your loop and you took a little sample of one colony from your spread plate and you put that onto your slant, remember that? That was a subculture technique. Mixed culture. Mixed culture contains two or more identified, easily differentiated species. And a contaminated culture is what none of us want, right? A contaminated culture is a culture that was once pure or mixed and now contains contaminants or unwanted microbes. Now, remember this, we need to know the metrics, scale of these organisms. The larger organisms are going to be your eukaryotic organisms, such as protozoa and yeast, followed by bacteria, followed by viruses. Remember, viruses are not alive, and they are so small that you cannot see them with the light microscope typically. You should know the term magnification. Magnification, where magnification is how many times larger your specimen appears than it really is. We talked about refraction. This is the bending or change in the angle of light as it passes through a medium, such as a lens. We talked about the first microscopes, such as Leeuwenhoek's pinhole microscope, being a simple magnifying lens with only a single magnifying lens. Later, compound microscopes added a second magnifying lens. Do you guys remember in the lab we have two sets of magnifying lenses, right? We have the ocular lens, or the eyepiece, that magnifies by 10x, and the objectives that rotate around, right? Those are 4x, 10x, 40x, and 100x. And those are the two sets. And remember, how do you get total magnification? How do you get total magnification of our microscope? You multiply the ocular, which is always 10x, by the objective, which you could switch from 4x, 10x, 40x, and 100x. You should know the parts of the microscope. So for instance, you should know that the eyepiece is called the ocular. It is 10x in magnification. You should know that the objectives can be swapped around. The smallest is the scanning objective lens, which is 4x. The next larger one is the 10x, or the low power objective lens. The next one is the 40x, or the high power objective lens. The next one is the 100x or the oil immersion lens. You should know that there's the, coarse adjustment knob, which is for adjustment with the 4x lens only, and the fine adjustment knob, which is used with the other objective lenses. You should know that the condenser, the job of the condenser is to focus a cone of light onto your specimen. The aperture diaphragm, also known as the iris, its job is to control the amount of light that goes through the condenser. And remember, that is a way of improving contrast, right? When you dim the iris, when you constrict that diaphragm, you improve the contrast of your specimen. Again, magnification, please know that these terms, magnification is how many times larger your specimen appears than it really is, while resolution is the ability to distinguish between two close small dots, and contrast is the ability to see an image as it stands out against the background. So how well your specimen stands out against the background. Remember that the specimen you see is an inverted and reversed, So your specimen is kind of upside down and flipped. You should know how to obtain total magnification. Remember, if you're using the 10x low power objective, your total magnification is 100x because you multiply it by the magnification of the ocular lens, which is 10x. If you're on the 40x objective, your total magnification is 400x. If you have oil with the 100x objective, then your total magnification is 1000x. Again, resolution is the capacity of an optical system to distinguish or separate two adjacent small points from one another. Remember, we talked about the resolution limit of the eye being 0.2 millimeters. All right. Now, we talked about the electromagnetic spectrum. Remember, I said that light consists of the spectrum between wavelengths 380 nanometers up to 750 nanometers. You should know that shorter wavelength light has more energy than longer wavelength light. And you should know that the shorter the wavelength of energy you use, the more resolution you obtain. And the longer the wavelength of the energy you use, the lower the resolution that you can obtain. And remember the oil immersion lens. What was the purpose of oil? You should know why we use oil. We use oil so we can collect more light into the objective by reducing reflection and refraction of the light. Again, the contrast. Contrast is the degree of contrast between a magnified image and its surroundings. Essentially, it's how much your specimen stands out against the background. Then we talked about different types of light microscopes. These are all types of light microscopes: bright field, dark field, phase contrast, interference, fluorescence, confocal, and super resolution light. The one we focus on within the lab is the bright field microscope. And do you remember the purpose of the dark field, phase contrast, and interference microscopes? These are better for contrast. These microscopes are better for contrast. We talked about wet mounts and hanging drop mounts. These are ways of preparing microscope specimens on microscope slides by adding a drop of sample and a cover slip. So a wet mount is probably the easiest. It consists of a drop or two of culture with a cover slip on top. Remember the disadvantages? The advantage was that it's quick and easy. The disadvantage was that you could damage larger cells. That cover slip smushes your cells. So instead, we use a hanging drop because a hanging drop uses a depression slide, which is kind of like a little swimming pool, to allow the cover slip to not smush your specimen. Do you remember we did a smear in the lab? We made a smear. You should know the purpose of making a smear, the steps to making a smear. Remember, to make a smear, you make a circle on the bottom side of your slide. Then you add a tiny drop of water to the slide. Then you add some bacteria and you spread it around the size of a quarter. And then you allow it to air dry. Remember, air dry. And then you heat fix. And you should know that heat fixing, the purpose of heat fixing. The purpose of heat fixing is to kill the cells, secure the specimen to the slide. Remember, adhere the bacteria to the slide, and it preserves cellular components with minimal distortion. Then staining smears. Remember, we use stain. And you remember the purpose of staining is to improve contrast. And we should know that basic or cationic dyes, basic dyes have a positive charge, so they're attracted to the acidic, negatively charged components of the bacterial cell wall. The cells are negatively charged. Basic dyes are positively charged. So for simple positive staining, we use basic cationic dyes. However, acidic or anionic dyes, these stain the background. Remember, they stain the glass slide. They are good for negative staining. So acidic, anionic dyes have a negative charge. They're repelled by acidic, negatively charged components of the bacterial cell walls. So these stains do not stain the cell. Instead, they stain the background for negative staining techniques. So again, positive stain uses basic dyes that stain the cell. Negative stain uses acidic dyes that stain the background. We did this in the lab. Remember, we saw positive simple staining with basic dyes such as crystal violet, methylene blue, safranin, malachite green. However, with negative dyes, or I should say acidic dyes, we stain the background. And remember, we used India ink as an example. Okay, simple staining. What does simple staining allow us to do? You should know that simple staining allows us to figure out the bacterial shape and arrangement, right? That's what simple staining is good for, shape and arrangement. Okay, and then we did differential stains. Remember, in the lab, we did differential staining, gram stain, capsule stain, right? These are differential stains that usually use two different colored stains so that you can see different types of cells. Remember with the gram stain, purple colored cells are gram positive, while pink colored cells are gram negative. With the acid fast stain, remember that these hot pink cells are acid fast. That means they have mycolic acids in their cell walls, while the blue cells are the non-acid fast cells. They do not have mycolic acids present in the cell wall. Then we did the endospore stain. Remember the endospore stains? You have the vegetative cell one color and the endospores another. Now, in our lab, the endospores were green and the vegetative cell was pink. But in this example from your book, the endospores are pink and the vegetative cell is blue. So, it's not a big deal because sometimes you use different dyes to accomplish the same thing. Then there are special stains. Remember, these are stains such as the capsule stain or the flagellar stain. Remember, some bacterium have a capsule, which is a type of glycocalyx. And by the capsule stain is a combination of an acidic dye and a basic dye. The basic dye stains the cell itself, while the acidic dye stains the background. And the only thing that's not stained is the capsule itself, which is waxy, which is nonpolar, so it does not cling to any of the charged dyes. Then there's the flagellar stain. Do you remember from lecture I had stated that flagella are too thin to see with the microscope? with the light microscope. So with the flagellar stain, you actually thicken up these flagella so that you can see the bacteria with the flagella. And that's it for chapter 3. Great. So let's take another little break time with Gizmo and Wicket and see what they're up to. We'll be right back for chapter 4. All right, welcome back. Let's carry on with chapter 4, Bacteria and Archaea. Remember that bacteria and archaea do not have a nucleus. Instead, they have nucleoid. Remember, I told you they have a single chromosome that's circularized. Unlike ours, we have linear chromosomes, and eukaryotes do have a nucleus, and they have numerous chromosomes. Bacteria and archaea, they do not have membrane-bound organelles. We talked about bacteria capable of doing all of the necessary activities of life, but also being able to form colonies, biofilms, and nanowires. Remember that bacteria, they vary in size, shape, and arrangement. You should know the 3 general shapes of bacteria. Coccus means spherical shaped, bacillus means rod shaped, and curved would be spirillum. Then pleomorphic bacteria do not have a particular shape, usually because they do not have cell walls. Mycoplasmas are an example of pleomorphic bacteria. Then we talked about arrangement. Remember, arrangement is how those shapes are paired up or lined up or how they're grouped up. So we have diplo. Diplo means pairs. Tetrads means groups of 4. Staphylo means irregular clusters. Strepto means chains. And diplo, strepto, palisades. Palisades are rows of side-by-side cells, side-by-side rod shapes in this case. Spirilla can be found in short chains as well. Biofilms, we should know that biofilms are microbial habitats with access to food, but they're basically a community. That means there's different microorganisms in a biofilm. Remember the biofilm formation, the steps to biofilm formation. First, the first colonists attach to a surface. Then they begin producing the biofilm material, this glycocalyx, this sticky extracellular sugar protein deposit, and then a community forms. This means different species live together inside of a biofilm. Next, we talked about different appendages of microorganisms, of these bacteria. So we have flagella and axial filaments for motility. We have fimbriae, pili, and nanowires for attachment points or channels. You should know the parts of a flagella. Remember a flagella? There's the basal body, which is embedded in the membrane of the cell. You have the hook, which connects the basal body to the filament, and the filament is the long whip-like structure. Do you remember that you can have different flagellar arrangement? So for example, you could have a monotrichous flagella, which means a single flagellum. Lopotrichous, meaning a tuft or a bunch and one spot. Amphitrichous, meaning a flagella at each end of the cell. Peritrichous, meaning completely surrounded by flagella. And do you remember the term for no flagella? That was atrichous, right? Atrichous. Then we talked about chemotaxis, which is movement in response to chemical signals. Positive chemotaxis means movement in the direction of a favorable chemical, such as sugar. And negative chemotaxis is movement away from a repellent or potentially harmful compound. Phototaxis is movement towards light. Now remember, there is runs and tumbles. Bacterium can run by counterclockwise. You should know that counterclockwise movement of the flagella allows the bacterium to run. And then when it switches to clockwise rotation, this results in a tumble. Remember that? So with bacteria, the flagella literally spins. Remember, we discussed that in the lecture, that with flagella in prokaryotes, the flagella literally spins. However, in eukaryotes, the flagella beats back and forth. Think of a sperm cell. Okay, and then do you recall an axial filament is a structure with an internalized flagella? This internal flagellum allows the microbe to twist, kind of like corkscrew motility. Then we talked about appendages for attachment or channel formation. The pilus, this provides adhesion but not locomotion. The fimbria or fimbriae provide adhesion. Remember, these were the short hairs. Then there was the flagella, which could also sometimes be used for attachment, and nanowires or nanotubes for, creating channels between bacterium. Here you can see the short fimbriae. Remember the short hairs? These are fimbriae for attachment. These are the fimbriae for attachment. Okay, small bristle-like fibers for attachment. Then there was the pili, and remember the process of conjugation I told you about. This is the sex pilus. It forms from one bacterium. It spans to another bacterium where it docks, and it undergoes this process of conjugation, which is a partial transfer of DNA from the donor cell to the recipient cell. Nanowires. Remember, nanowires, very thin, long tubular extensions of cytoplasmic membrane that bacteria use as channels. They can transfer amino acids and electrons through these channels. Then we talked about surface coatings, the S layer and the glycocalyx. The S layer is thousands of copies of a single protein, providing protection from the environment on the surface of the cell. And there were two forms of glycocalyx, the slime layer and the capsule. Remember the glycocalyx polysaccharide units, which may or may not include protein. The slime layer, which is loosely associated with the cell. Remember, the slime layer is loosely associated with the cell, while the capsule is more tightly associated with the cell. Here you can see a capsule versus a slime layer. Here you can see a capsule with the capsule stain. Capsules. Remember, pathogenic bacterium tend to form capsules, and these protect the bacteria against your immune system. They protect against phagocytic white blood cells of your immune system. And we talked about how slime layers aid in attachment as well as resisting drying out. Next, we talked about the cell envelope, right? On the surface of the cell, we've got the cell wall, the plasma membrane, and then sometimes you have an outer membrane, right? In the gram-negative bacterium have an outer membrane. So let's talk about this. The Gram stain, developed by Hans Christian Gram. This delineates between two types of bacteria, right? The gram-positive bacteria, remember, that have a thick cell wall of peptidoglycan, and the gram-negative cells, which have a thin cell wall of peptidoglycan, but they do have an outer membrane as well. Do you remember this? We need to know this. Here on the left, you have a gram-positive cell wall architecture. You should know that you have your plasma membrane, and then outside of that, you have this thick layer of peptidoglycan. You also have these structures called lipoteichoic and regular teichoic acids. Okay, whereas with the gram-negative cell wall on the right side, you have the plasma membrane. Then you have a very thin layer of peptidoglycan, and you have an additional outer membrane. And do you remember the important components of the outer membrane? we said you have the outer membrane, then you have these big porins, which form pores in the outer membrane. And do you remember these pink structures? These are not phospholipids in pink. These are phospholipids down here in kind of orange. But these pink structures are not phospholipids. Remember, they are LPS, lipopolysaccharide. Here, lipopolysaccharides only in gram-negative cells. Remember that? So again, peptidoglycan makes up the cell walls of bacteria, but it's unique to only bacteria. This means that archaea do not have cell walls of peptidoglycan, and eukarya and archaea, they do not have peptidoglycan cell walls. Only the domain bacteria has the peptidoglycan cell walls. And here you can see that the peptidoglycan is part sugar and part protein. And remember the gram-positive cell wall? What do you need to know about that? It includes teichoic acids, lipoteichoic acids, which help to, maintain the cell wall and provide an acidic charge on the cell surface. But essentially, what is a gram-positive cell wall? You have your plasma membrane or cytoplasmic membrane, and you have a thick layer of peptidoglycan with those lipoteichoic acids. Okay, that's what we have in a gram-positive cell wall. But a gram-negative cell wall, remember, this is different. Gram-negative cell wall, you have the cytoplasmic membrane, this plasma membrane. Outside of that, you have a thin layer of peptidoglycan. And then outside of that, you have a whole outer membrane. Remember the outer membrane? And do you remember these structures here are called porins? And then these pink lipids are known as LPS, or lipopolysaccharide. Here you go, lipopolysaccharides. And do you remember the importance of lipopolysaccharides? Do you remember? Lipopolysaccharides contain, I just want to show you this. I'm skipping ahead real quick. Lipopolysaccharides contain this important structure called lipid A. Remember why we discussed why gram-negatives are important? Because they have lipid A as part of their outer membrane, LPS, is because lipid A is highly toxic to our bodies. If you get lipid A in your system, it can cause systemic problems, right? And it can even cause fever and shock, right? Septic shock. So it's important to know that lipid A is a component of LPS, and LPS is a component of the outer membrane of gram-negatives. So you should know that gram-positives should not have lipid A. They should not have LPS because they don't have an outer membrane, right? Awesome. Okay, so let's continue on with the non-typical cell walls. Remember, we mentioned in lab that some bacterium are known as the acid-fast bacterium, and that's because the lack of cell wall structure of gram-positive or gram-negative bacteria. Instead, they have these mycolic acids, also known as cord factor, in their cell wall. And this is a fatty acid, so it's non-polar, and that's why it doesn't stain, very easily. Then you have your acid-fast stain used to show if bacterium have this mycolic acid or not. And then mycoplasmas, remember the mycoplasmas, which are pleomorphic because they don't have a cell wall. But most bacteria do have a cell wall, right? Most bacteria are gram-positive or gram-negative. But the weird ones, they could be acid-fast because they have the mycolic acids, or they could have no cell wall, which is weird, and they're pleomorphic. They don't have a particular shape. Okay. And then we have the cytoplasmic membrane. Remember the cytoplasmic membrane, which is there for regulating how much, transport into the cell and out of the cell. The cytoplasmic membrane is selectively permeable. And what is the function of the cytoplasmic membrane? Energy reactions, nutrient processing, synthesis, and transport. But the main function of the membrane is selective permeability, allowing certain things into the cell and certain things out. And again, do you remember that gram-negative outer membranes could include what? They include LPS, which has this toxic endotoxin, lipid A. You should know that lipid A is called endotoxin. And the outer membrane of gram-negatives also contains those porin proteins, which allow small molecules to penetrate the outer membrane. The cytoplasm. The cytoplasm is the gelatinous solution contained in the cytoplasmic membrane. This is found in all cells. All cells have a cytoplasm. Bacterial chromosomes. Remember, there's a single chromosome that's circularized. Remember that? And it's called the nucleoid. Then you could also have plasmids, which are non-essential pieces of DNA, small double-stranded circles of DNA that give important genes, important traits to the cell. Ribosomes. Do you guys remember that ribosomes are non-membrane bound organelles found in all cells? So prokaryotes and eukaryotes have ribosomes, and ribosomes synthesize proteins. And do you remember there's the small subunit of the ribosome and the large subunit of the ribosome? They kind of come together like a hamburger bun, right? The hamburger bun has the large bun and the small bun. And do you remember this is important for synthesis of proteins, right? Ribosomes are involved in synthesis of proteins. And do you remember that bacterial ribosomes are much smaller? They're called the 70S ribosomes. And eukaryotic ribosomes are larger. They are the 80S ribosomes. Next, we learned about inclusion bodies, which are little areas of storage, right? Storage sites inside of prokaryotes. And that prokaryotes have a cytoskeleton, a protein structural support system inside of the cell. Next, we learned about endospores. Remember, endospores, they are defensive structures which withstand hostile conditions and facilitate survival. Remember, only 2 genus of bacterium can form endospores, only the Clostridium and the Bacillus. There's the vegetative cell, which is metabolically active. This is also known as the mother cell that grows. And then the endospore itself, which is this inert, resting, dormant spore, which is designed to withstand those harsh conditions. Sporulation is the process of forming a spore, and germination is the process of the spore, hatching, if you will, it growing back into a vegetative cell. An endospore can resist heating, drying, freezing, radiation, chemicals. Remember that the endospore forms mainly when there's a depletion of nutrients. And the sporangium is the sporulating cell. Remember the steps to sporulation? Please learn the steps, these 9 steps to sporulation. I might ask you about these steps. How a bacterium forms an endospore. Please learn the steps involved in sporulation. And then there was germination, right? When the favorable conditions come back, we have germination. The spore germinates. It grows out back into a vegetative cell. Okay, we should know that there are certain diseases caused by spore formers. So you should know that Bacillus anthracis causes the disease anthrax. Clostridium tetani causes the disease tetanus and lockjaw. Clostridium perfringens causes gas gangrene. And Clostridium botulinum causes botulism. We should know these diseases caused by common spore formers. And do you remember another one we discussed in the class? Clostridium difficile, and how it causes, C. diff. It causes the colitis. And then lastly, we talked a little bit about archaea. Not a lot, but we talked a little bit about archaea. And remember, I really need you to know that archaea are more closely related to the domain Eukarya than they are to the fellow prokaryotic bacteria. All right. And what else did I need you to know about archaea? They are prokaryotic, and they are found in extreme environments. Hot, cold, salty, acidic, basic, pressurized. So they are found in extreme environments. And remember, they do not have peptidoglycan cell walls. Archaea do not possess cell walls of peptidoglycan. And then we also talked about bacterial species, which are based on traits that they share. So how do you know that two bacterium are of the same species? They have similar traits. And that's it. That's it for chapter 4. A lot of nice meaty material there in chapter 4. Good stuff to learn. So hopefully that was a good little review. Let's take our final break time with Gizmo and Wicket. Come back strong with chapter 5 so we can finish off this review for exam 1. All right, let's do it. All right, welcome back from break time. Let's keep going with chapter 5, eukaryotic cells and microorganisms. All right, so let's carry on. You should know, again, any term in bold, organelles. What are organelles? Remember the endosymbiont theory. So this was interesting. It explained the origin of the mitochondria and chloroplast. You should know that the endosymbiont theory states that the mitochondria and the chloroplasts came from bacteria once upon a time. They used to be bacterium, which were kind of endocytosed into the early eukaryotic progenitor. And how do we know this? They have their own circularized DNA. They have 70S ribosomes, and they have their own 2-layer membranes. So please know what the endosymbiotic theory states. You should know that these are the eukaryotic organisms studied by microbiologists, so protozoa, fungi, algae, helminths. You should know these are the structures found in all eukaryotic cells, while these are the structures found in some eukaryotic cells. So for example, I might say, which of the following is not found in all eukaryotic cells, right? So think about that. Okay, flagella. You should know that eukaryotic flagella are much different than prokaryotic flagella. Remember, they have the beating, the back and forth beating. They're much thicker and more complex. You should know that cilia are much thinner, smaller components. They have a power stroke motility, which means there's a power stroke followed by a recovery stroke. Power stroke followed by a recovery stroke. And there are these fine, small hairs that are found on eukaryotic organisms for motility. Then we have the glycocalyx, which also called an extracellular matrix. A cell wall. Remember that not all eukaryotes possess a cell wall. So, for example, fungi have a cell wall of chitin. There is cellulose found in cell walls of different types of algae. Diatoms have cell walls of silica, but not all microorganisms possess a cell wall. Not all eukaryotes possess a cell wall. Then there's the cytoplasmic membrane, which can contain sterols to stabilize the eukaryotic membrane. It's selectively permeable. That's the main job of the membrane. All right, moving on. Remember the kingdom fungi, very important kingdom fungi. There are macroscopic fungi, including mushrooms, puffballs, gills fungi. And then there's the microscopic fungi, molds, and yeast. We should know that yeast are a type of fungi which have round to oval shaped cells, while mold have hyphal cells. Mold have cells that are thread-like or hyphal. Here's the yeast cells. I said the yeast cells are semicircular or oblong in shape. And you should know that yeast divide by a process known as budding. You should know that fungi obtain their nutrition as saprobes. Saprobes obtain their nutrition from dead organic matter, dead plants, dead animals. Or remember, some fungi are also parasites, which means they can feed off of living animals or plants as a parasite. Remember that mold form mycelium. Mycelium is the woven, intertwining mass of hyphae. That's that fuzzy layer that appears on your fruit when you have a mold, growing on there. And you should know that the fungal spores are not endospores. These are not so resistant as endospores. They're just there for reproduction. They are asexual spores for dissemination. You should know the difference between sporangiospores and conidiospores, also known as conidia. Sporangiospores are spores inside of a cap, while conidiospores or conidia are spores not within a cap. And then you should also know that sometimes these fungi prefer to do sexual reproduction, and this is so that they have potentially advantageous traits for adaptation. Here on the left, you see the sporangiospores inside of the cap, and on the right, you see the conidiospores or conidia outside of the cap. Again, you have pathogenic fungi, which can cause disease, which acquire their nutrition by feeding off of living animals and plants. Okay. You should know that fungi have an essential role in decomposing organic matter. They can, so decomposing organic matter is essential in the ecosystem for returning essential minerals to the soil. That beneficial fungi can form associations with plant roots that increase water and nutrient absorption. That beneficial fungi can give us antibiotics, they can give us alcohol, organic acids, and vitamins. They can provide us with food flavoring. And like I said, they can be used in the production of beer and wine, and they can be used in the production of bread as well. So these are some beneficial impacts of fungi. Next, moving on to the protists. Remember, the protists are algae and protozoa. Algae and protozoa are single cell eukaryotes known as protists. So a protist is any eukaryotic unicellular or colonial organism that lacks true tissues. These include the seaweed and the kelp, which are types of algae, as well as the green algae, right? The green algae. And the diatoms, which do photosynthesis, right? So kelp, seaweed, green algae, diatoms, they all do photosynthesis, very important organisms. They make up plankton. Remember plankton at the bottom of the food chain. They make so much oxygen for us to breathe, 70% of the Earth's oxygen for us to breathe as well. So now moving on to the protozoa, which include about 65,000 different species. You can find these in moist habitats, including fresh water and soil, but there can also be parasitic forms living with plants and animals. They can have structures analogous to mouths, digestive systems, reproductive tracts, or even legs. There are free living species and parasitic species, but overall they are heterotrophic, which means they require their food in complex organic form. So they can't make their own energy, right? They can't photosynthesize. They can move. Most of them have some form of locomotion. They can move with pseudopods or false feet. think about amoeba. They can move with flagella. They can move with cilia. They have different stages of life, right? The life cycle. They have the trophozoite or motile feeding stage, which is active, and then the dormant cyst stage, which is a resting stage. We should know that some of these protozoa are important pathogens. And here's some examples. Entamoeba histolytica, which is a type of amoeba. It causes amoebiasis, or an intestinal problem, intestinal disease. Naegleria, which causes a brain infection. Remember, we talked about Naegleria fowleri, found in the lakes around Texas, which can cause the brain-eating amoeba, the brain infection. Balantidium coli, or B. coli, which can cause balantidiosis, which is an intestinal disease. Giardia, this is another interesting one. Giardia lamblia, which causes giardiasis, which is a intestinal disease, also known as traveler's diarrhea. Okay, those are ones to know about. Here's some pathogenic flagellated protozoa, the trypanosomes. Trypanosomes are flagellated protozoa. Trypanosoma brucei, which causes African sleeping sickness. Trypanosoma cruzi, which causes Chagas disease. These are important parasites of humans. Move on to the helminths. Helminths are worms, right? We have flatworms and we have roundworms. The flatworms are thin, often segmented worms. They're divided into two groups, the cestodes, which are the tapeworms, and the trematodes, which are the flukes. You should know that cestodes are the tapeworms, trematodes are the flukes. However, the roundworms are known as nematodes. You should know that roundworms are the nematodes. And yes, we do study worms in microbiology because oftentimes you are infected by the microscopic eggs from undercooked meat or by contaminated water or something like that. We talked about the life cycle of the worm, the fertilized egg, followed by the larval stage, followed by the adult stage. Nematodes, or roundworms, they have sexes with different morphologies, so they have a clear male and female sex. Trematodes have sexes of separate or male and female sex organs in the same worm. They can also be hermaphroditic, while cestodes are generally hermaphroditic. Okay, and how does the life cycle work? Transmission of an egg or larva to the body of another host, either a different or the same species, then intermediate or secondary host in which the larval development occurs, and the definitive host, the host in which adulthood and mating occur. And what are the routes of infection? Oral intake or penetration of unbroken skin. Humans are the definitive hosts for many species of the sole biological reservoir for about half of the diseases. Animals or insect vectors also serve as reservoirs. Next, we explored a helminth cycle, the pinworm. Enterobius vermicularis causes a very common infestation of the large intestine, the pinworm infection. Worms range from 2 to 12 millimeters long. They are tapered, curved, cylinder-shaped. Simple, uncomplicated infection that does not spread beyond the intestine. Remember how it works? The worms form and eggs emerge from the anus. Scratching and contaminates the hands with the fertile egg. And then by touching the mouth, you swallow or self-infect with the larva, or with the eggs, I should say. Then the eggs can be transferred to a new host as well through contact. and then they develop and grow within the intestines. All right. And that's it. That's it for chapter 5. Those are the important concepts from chapter 5 I wanted you to know. Hopefully this was a good review, or at least get you started on your way studying for exam 1. I wish you luck. I know you can do it. Study hard. Again, please study all the slides, not just what I highlighted in this review, for best results, right? So do your best, study hard, learn the material, and you should do just fine on exam 1. I wish you well.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.